Hi everyone. In the immunology subject, we are going to learn about cells of the immune system. Yes, in our body or a human body contains a certain type of the cells which are protecting us from different types of infectious agents like microorganisms, bacteria, virus, etc. And those cells are all being called or classified as cells of the immune system. Examples of these cells of the immune system are lymphocytes, monocytes, macrophages, neutrophils, eosinophils, null cells, etc. Okay, and in the previous part of the cells of immune system, we had gone through the different lineages of these stem cells into lymphoidal lineage, then myeloid lineage, and then erythroid lineage. And we in detail discussed about this lymphoidal lineage in the part 1. You can go through it. And here in this part, we are going to discuss majorly about the cells that are arising from myeloid progenitor or myeloid lineage. So here we are going to have the different types of the cells. Those are going to be the monocytes, then the granulocytes, and the accessory cells. Okay, so these are all going to be the cells that are coming from the lineage of myeloid progenitor. So let's see this myeloid lineage, and then what are the types of the cells we involve in it, and what are the functions of them. Okay, let's begin. So here, coming to the cells of myeloid lineage, the myeloid lineage includes cells involved in the process of especially phagocytosis, and the other cells like accessory cells too. So that means the cells both of having the nature of either the mononuclear or the polymorpho, both are going to have the phagocytic in nature. And along with these two systems, we are also having the accessory cells. Those are being called as mast cells, platelet cells, and antigen presenting cells. So the mononuclear phagocytes are giving rise to the monocytes, and those are getting modified into the macrophages in the tissues. And the polymorphonuclear, the nucleus is getting modified. That means it can be of a single lobe or bilobe or a multi-lobe. The nucleus is going to be folded in different shapes. So that's why these cells are going to be called as polymorphonuclear leukocytes. As these are a type of WBC, we call them as leukocytes. So in the immunology of the cells of immune system, we are mainly concerned about these cells here. So let's begin with the mononuclear phagocytes or monocytes. So monocytes are the circulating immature cells of the mononuclear phagocytic system. And these cells are going to be of having very large in size. They are motile, amoeboid, phagocytic with a single nucleus. So here in the function of, so this is how a typical macrophage is going to have a shape. It is not going to have a proper shape. Okay, and that's why we call it as amoeboid. And as an amoeba, it is going to possess the pseudopodia, all these things. And here, as we are discussing about the monocytes or large motile means with the pseudopodia, they are going to have the swarming motion, and amoeboid phagocytic with a single nucleus. That's important here. There are about 2 to 10% of the total WBC count in a man. So suppose if we are having the 100 WBC, that is the white blood cells, out of which 2 to 10 number will be the monocytes. And any cell in the body is going to be derived from the bone marrow only. In the same manner, the monocyte is also derived from the bone marrow. But the cells that was derived from the bone marrow are going to be called as promonocyte. When they enter into the bloodstream, and we call them as monocytes. And these monocyte form is going to be of very short time unless it is going to be in the circulation of the blood. But once these monocytes migrate into the tissues or organs, then they are going to be becoming as macrophages. Until the monocytes are going to be called, are named as monocytes when they are going to be in the blood circulation. But these monocytes, when they migrate into the tissues, we call them as macrophages. Yes, macrophages, as I told you, the monocytes of the blood migrate into the tissues and differentiate into the macrophages. And these macrophages are having a very long lifespan. That means they can live for a longer time. That means from few months to even the years also. So that's the reason why they are going to act as a main soldiers for us from protecting the pathogens. And not only that, they are present everywhere in the body. The spleen and liver contains more macrophages. So wherever you are going to have the more exposure of these chances of exposing to the antigens, there these are going to be more in number. The spleen, we know that it is going to filter our blood, which is one of the main source of circulation throughout the body. And these monocytes, when transformed into macrophages with morphological and functional features, depending upon the type of tissue, they are going to be named differently. That means these macrophages, when they are going to be in the different kinds of the organs or tissues, they are going to be called with some other name. So what are those names? Let's have a look of these all names. For example, if the macrophages are going to be present in the lymph nodes, they are going to be called as dendritic macrophages. In the same manner, if the macrophages are going to be present in the lungs, that means from the blood circulation, the monocytes, when they enter into the lungs, now the lungs containing these macrophages are called as alveolar macrophages. In the same manner, the macrophages that are present in the liver are called as Kupffer, and the macrophages that are present in the bone are called as osteoclasts, and the macrophages that are present in the kidney are called as mesangial cells, and the macrophages that are present in nervous system are going to be called as microglial cells, that means brain cells, and then the one which is present in the connective tissue are going to be called as histocytes. So these are all macrophages only, but they are going to be called with the different names because they are present in the different organs and tissues. So this is all maybe for the convenient of identifying the cells. Okay, but what are these are all cells? Nothing but the macrophages only. Okay, let's see what are the functions of these macrophages, which is very important of reading about this one. So majorly we are having the four types of the functions that can be seen by these macrophages. The first one is phagocytosis, very important for the short note also. Then processing of antigen, then antigen presentation, and then some sort of a secretory function. So altogether we are going to discuss about the four types of the functions of macrophages. So let's see the first one, phagocytosis, engulfing. So it is going to engulf something. What is that something? Nothing but the viable microorganisms or dead cells or debris. So these are going to be engulfed by our macrophages and they are being broken down. So such type of the mechanism is going to be called as phagocytosis. So let's see the process that is occurring in the macrophages of this phagocytosis. So the macrophages are going to be strongly phagocytic to a wide variety of particulate materials such as the viable microorganisms, dead cells, debris, etc. So what is the process of this phagocytosis that is occurring in the macrophage? So this is the figure or diagrammatical representation of the process of phagocytosis. So let's see this one. So in the first step of phagocytosis, macrophages are going to be attracted by these antigens and move toward these antigens, or a variety of substances are going to be generated in an immune response process. And this attraction because of release of some chemicals is going to be called as chemotaxis. What we call it as chemotaxis. So that is the first step, attracting with the release of some chemicals, and these macrophages are moved towards them, and that process is going to be called as chemotaxis. Then coming to the next step after chemotaxis is the attachment of these antigen to the membrane of this macrophage. So slowly what happens? It tries to attach to the macrophage membrane. And this attachment is going to be differed depending upon the complexity of the pathogen. That means if the whole bacteria or the whole virus particles comes and attach to it, then the phagocytosis action will be very good. In the same manner, if the complexity of the antigen, that means only a part or some sort of light proteins are going to, that is light part of antigens are going to come, then it is going to have a lowered or poor or less readily phagocytosed. Okay, so that's the only condition that it is going to be there. You can neglect it. So what is the next step? It is going to, that means the antigen binds to the membrane of macrophage. Now after the attachment or adherence of the antigen, there occurs some sort of a protrusions from this membrane of the macrophage. So you can see some sort of extensions surrounding this antigen without touching the antigen. Okay, so that is what happening. Now these extensions are being called as pseudopodia. Now these pseudopodia encloses the material, that is the antigen, within a membrane bound structure called as phagosome. Now it has formed the sac-like structure and it was dragged to inside, and that sac-like structure is called as phagosome. And this phagosome which enters the cell will enter a pathway called as endocytic processing pathway. So inside it is going to be lysed, that's why it is called as endocytosis. Now once this phagosome enters the cell, immediately it is going to combine with another cell called as lysosome. So now a combination of this phagosome and the lysosome together, we call it as phagolysosome. So here is a place where the lysosome is going to release some sort of enzymes called as lysozyme and hydrolytic enzymes that digest this antigen or the material that is present in this phagolysosome. So once that antigen is going to be lysed or digested, obviously it is going to be thrown outside this macrophage, and that process is going to be called as exocytosis. So that was thrown out of the digested contents of the phagolysosome or then eliminated in a process called exocytosis. So this is the whole process of the phagocytosis mechanism that is occurring in the macrophage cells. So this is the first function, which is very important. Then moving to the second function of macrophage is processing of antigen. So what is a processing? That means if you are getting any product, it is going to have some sort of a processing step such that its quality or appearance is going to be enriched. In the same manner, here the macrophages are going to process the antigens that are entering to the body, and they are going to be presented. First of all, it is going to be processed, and then after the processing, that means they are increasing the immunogenicity of an antigen, and then they are going to be exposed or presented to the T cells, where these T cells will combine with them and they are going to have the removal from the body and making us to be immunized. So this is the second step where it is presenting. First, it is going to process the antigen. So you can see here the macrophages are involved in the processing of antigen before they are presented to the T and B cells. But the steps involved in this process is not that much clear. Okay, and here once they are going to be presented to T cells and B cells, obviously cell mediated immunity and antibody mediated immunity increases with the processing of antigens. And coming to the third type of the function, that is antigen presentation. After the processing, it have to present it. To whom it is going to present? That means the macrophage is going to present the antigen to T cells and B cells. So it is going to present to the T cells, which are going to have the T helper cells and T cytotoxic cells. So why it is going to present the antigens to these two cells means they possess some sort of a determinants called as MHC determinant of class 2 on the surface, where a macrophage can recognize them and it is going to present this antigen. Suppose the T cells are not going to have these markers, surface markers, then the macrophage cannot cooperate with T cells and cannot present the antigen to them. So where there will be no question of cell mediated immunity. Okay, so this is how it is going to present the antigen. And then coming to the fourth type, secretions. So these macrophages are also going to secrete a number of substances such as components of complement system, then hydrolytic enzymes, some toxic forms of oxygen, and even the interleukins. So this is how these are also going to be involved in protecting of a human body from the different types of antigens or infectious agent. So this is the first type of a system that we have discussed is macrophages, mononuclear phagocytic cells. Then moving to the second type, that is polymorphonuclear leukocytes or granulocytes. So these polymorphonuclear leukocytes were named so because of having the different shapes of the nucleus. Only one nucleus is there, but it is going to possess the different shapes. Like this, you can see here, this is one shape. It's a bilobed shape, and here it is going to have a multilobed shapes. Okay, so this is how, if a nucleus is going to have the different morpho, that is shape, then we call them as polymorpholeukocytes. And as they possess the granules in the cytoplasm, these are also going to be called as granulocytes. Okay, and the lobes, that means the morpho, when the nucleus is going to be of different shapes, then there may be of 2 to 5 lobes are going to be formed. So on the basis of this cytoplasmic granule staining, these are going to be classified into 3 types. The first one is basophil, then eosinophil, and then neutrophil. So you can look at these pictures, then you can have a clear idea about the structure of these cells. So this is a neutrophil structure, and this is an eosinophil structure, and this is a basophil structure. So let's start with the basophil. Okay, the granulocytes, which are going to have the cytoplasmic granules staining bluish black with basic dyes, are going to be called as basophils. So that means the granulocytes, when stained with a basic dye, if these cytoplasmic granules are going to show the bluish black color or stained with the bluish black color, then those cells are going to be called as basophils. And these basophils are going to be of about only 2% of the total WBC count. Suppose if 100 cells are there, out of which only 2 are going to be of our basophils. And these basophils, obviously you can see they don't have the proper shape, they are irregular shape, and the nucleus is going to be off with 2 lobes. So here you can see the 2 lobes. And the basophils are going to possess some sort of a chemical substances called as a histamine, serotonin, etc. in these granules. Okay, and these are going to be released after stimulation of antigens. So after once the antigen enters and they stimulate these cells, so obviously these granules are going to release some sort of a chemicals called as histamine or serotonin. And even these basophils are going to have another important feature, that is possessing of IgE receptors. So that receptor, IgE receptors are going to have a very prominent role in allergic reactions, that is hypersensitivity reactions. So that we will discuss in another part. So overall, the basophils are going to be of containing the bluish black granules, and these are about only 2% of total WBC count, and the nucleus is going to have the 2 lobes, that is bilobed structure, and these are going to possess a special type of chemical substances called as histamines and serotonin. Then moving to the second type of granulocyte, that is eosinophil. So here is the eosinophil, and as the granules are going to be seen in the red color. So how they are going to be given as a red color means the granules when stained with an acidic dye, that is a eosin. If the granules are going to be stained with color red, then we call them as, or we assign them as eosinophils. And these are going to be of about only 2.5% of the total WBC count. And eosinophils are going to have a very major role in defense against mainly of our parasitic organisms like protozoans, helminths, okay? And they are going to fight against these parasites by secreting some sort of histamines, aryl sulfatase, and other cationic proteins which damage plasma membrane of the parasitic organisms. So that's how they show the chemotoxicity. That means the chemicals that are released by these cells are going to show the toxic nature on the protozoan parasites. That's how we call it as that property as chemotoxicity. And these eosinophils count will be more in number during the allergic reactions. Okay, so that state or condition is going to be called as eosinophilia. And then coming to the third type of granulocytes, that is neutrophils. So here the name itself is saying neutral. So it is not neither having a red color nor a blue color. So such type of the cells or granulocytes are going to be termed as neutrophils. So these are the cells containing cytoplasmic granules which do not take up acidic or basic strains strongly. Hence these are called as neutrophils. And if you observe here, the nucleus is going to have the multi-lobe. It's not bilobe, it is a multi-lobe. Several lobes are there. So here you can see. Okay. So multi-lobe structure. And here is a bilobe structure, eosinophil, where it is going to be of multi-lobe structure. And these cells are going to have about 10 to 20 micrometers in diameter. And one more important point here is these neutrophils are going to be of more in number when compared to the eosinophils and neutrophils. For example, in the total WBC count, these neutrophils are going to be of 60 to 70%. 60 to 70%, whereas your eosinophil and the basophil are only 2% and 2.5%. But here, these neutrophils are 60 to 70% of total WBC count. And the major function of these neutrophils is phagocytosis. Like how the macrophages are involved in the phagocytosis, here the neutrophils are also going to be involved in the phagocytosis process. Whereas the eosinophil and the basophil are involved in the chemotoxicity. But here it is involved in the phagocytosis. So these neutrophils can attack and destroy bacteria and viruses in the circulating body. So these neutrophils reach the affected area along with the macrophages. That means they accompany with the macrophages and destroys the unwanted or foreign antigen by the process called as phagocytosis. So that's how the neutrophils are going to be more in number along with the macrophages, which are playing a faster defense of removing the antigen from the body, and that's how providing the immunity to the host. So these are the different types of the cells that are involved in our immunity, and that's how we finish all the types of cells of immunity. In the previous part, that is in another part, we had gone with the lymphoidal lineage, that is lymphocytes, the different types of lymphocytes, and in this part we had gone with the myeloid lineage. So lymphoidal lineage, lymphocytes are of three types: T lymphocytes, B lymphocytes, and null cells. Okay, and the null cells are again of two types: natural killer cells and the killer cells. So this whole thing we have discussed in detail in other part, and here in this part we had gone with the monocytes, that is macrophages, their function, then polymorphonucleosides, eosinophils, basophils, and the neutrophils. So that's end of this cells of the immune system. Thank you.